This is Chenye Khoto and welcome to my YouTube channel. I'm going to be presenting a presentation for my assignment of BPT Becoming a Professional Teacher 1501, and with me here I'm going to be using demonstration to facilitate a lesson for the grade 12s where I'll be talking about meiosis. With me here I've got some keywords where I'll be talking about a chromosome which contains the centromere, the gene, and the chromatids. We've got all as well the nuclear membrane, which is the spaces in between here, the spindle fibers, which are the dotted lines, as well as the pole. In meiosis, we've got the interface where the chromatid network replicates and doubles itself. The DNA replication takes place to double the amount of the genetic material, and the single strand of the chromosome becomes double-stranded. In the process of meiosis, we've got the first meiotic division, in which we are having the first prophase, which is the early prophase. And in early prophase, we've got the visible chromosomes that are formed by the chromatids right here, as you can see. And then the homologous pair lie in between these poles. And in the late prophase, we're going to be having the chromosomes attached to the spindle fiber by the centromere and the nuclear membrane right here. As you can see, there's a nuclear membrane. In this late stage, the nuclear membrane starts to disappear. And when prophase 2 takes place, that is where crossing over occurs. And when crossing over occurs, the chromosome touch at the chiasmata and the homologous pairs joins. And then we've got a metaphase in which the homologous pairs lines up at the equator of the cell, and we've got the anaphase in which the spindle fibers contract each other and pull each homologous pair towards the pole, and the homologous pairs pulled towards the chromosome move to the opposite poles. And in telophase, that is where we'll be having a complete set of chromosomes, each in a pole, and the two new forms will be celled. The two new cells will be formed, and each cell will have a set of haploid chromosome. And then we've got the second meiotic process, or the second meiotic division, in which we'll be having the second prophase, where the spindle fiber will be forming between each poles, and each chromatid contains of a new chromatid joined by the centromeres. As you can see, this is where crossing over took place. And then in metaphase 2, the chromosomes lines at the equator of each cell, and in anaphase, the centromeres starts to split and the chromatids move towards each pole. And in telophase, we've got the chromatids now called the daughter chromosomes, and the nuclear membrane reforms again to form in each new cell. And then when telophase has taken place, that is where a process of cytokinesis takes place. And cytokinesis takes place when two new daughter cells are formed from each parent, and the nucleus of each daughter cell has now haploid number of chromosomes. I obtained all this information from grade 12 life science learners books, life science learner books, and the study guide of understanding. Thank you all for watching. Thank you.